Thank you, Dr. Shnaik. Good morning, everyone. Uh, it's a little cold out here, but I think it's a warm welcome to all students, scholars, faculty members of various colleges, universities, and institutes. My name is Dr. Rohit Sharma, academic editor in MycoIndia. It's my pleasure to welcome you all to the international seminar on recent developments in fungal biotechnology and informatics, which is jointly organized by Association of Fungal Biologists, MycoIndia, and MycoAsia. I also welcome members of Association of Fungal Biologists, Team MycoIndia, and MycoAsia, especially Dr. Sunil K. Deshmukh, the President of the Association, Dr. Shashi Rekha Suresh Kumar, the Secretary of Association of Fungal Biologists, Dr. Mahavir Gosavi, the Treasurer, Dr. Bale Damodar Shenoy, Founder MycoIndia and Co-founder MycoAsia. I also extend my warm welcome to all the mentors and other members of the Association of Fungal Biologists, as well as those who are associated with MycoIndia and MycoAsia. Professor D.J. Bhatt sir, Professor Sridhar sir, Dr. Ravindra Bagul sir, Dr. Sanjay Saxena, Dr. Vineet Meshram, Dr. Nishita, Dr. Malika Sutar, Dr. Ral Sahab Yadav, Dr. Kamlesh Shukla, Dr. Lokesh Gambhir, and many more. Now I invite Dr. Shenoy, the Principal Scientist, CSIR National Institute of Oceanography, Visakhapatnam, to give a brief introduction of the genesis of today's international online seminar and also the plan of today's event. Dr. Shenoy.
Thank you, Dr. Rohit. I hope I am audible.
Yes, yeah, very clear.
Respected Dr. Sunil Deshmukh ji, President, Association of Fungal Biologists, India. Ladies and gentlemen, members of the MycoAsia, members of the MycoIndia, members of the Association of Fungal Biologists, ladies and gentlemen in the audience, thank you very much for joining in today. First of all, I would like to extend my gratitude to Dr. Deshmukh and the Association of Fungal Biologists for associating with MycoAsia and MycoIndia for this event. I think this idea came up when we met Dr. Deshmukh and Dr. Shashi Rekha and Dr. Mahavir and other members of the esteemed members of the AFB at Mumbai for the first annual meeting of AFB. There we thought we could come together and conduct online programs for the benefit of budding mycologists of the world. I mean, by the help of this online platform, we can reach the audience from all parts of the world. That is the idea. And it was Dr. Deshmukh actually who was instrumental in getting the wonderful speakers today. And he is the first speaker. He'll talk about coloring metals, materials from fungi. Second, Dr. Bhupesh Taneja from IGIB, CSIR IGIB, New Delhi. He will talk about his informatics work on dermatophytes actually. Then Dr. Usha will talk about cosmeticals, and then afternoon Ms. Mona Lausani will talk about mycomaterial. At the end we'll have Dr. Florenca from Spain. She will give she will share her experiences working with fungi in the industrial setup. This is the plan. We plan to start in next 1 or 2 minutes after the brief introduction of Dr. Deshmukh to the audience. That will be done by Rohit Sharma. Then we hope to finish this today's session by 4:30 Indian Standard Time. Okay. This is from my side. Thank you very much, Dr. Rohit, for agreeing to be the face of today's event. As usual, you are always wonderful to conduct this program. Over to you, Dr. Rohit.
Thank you, Dr. Shenoy. Our today's first speaker, Dr. Sunil K. Deshmukh, is a renowned figure in mycology and scientific research with a career that has profoundly impacted the field. beginning with a PhD from Dr. H.S. Gour University in 1983. His academic foundation propelled him into becoming an expert in industrial mycology. His tenure at companies like Hoechst Marion Roussel Limited and Piramal Enterprises Limited in Mumbai marked a significant advancement in mycology's industrial applications, especially in the drug discovery. Dr. Deshmukh played a pivotal role in developing microbial culture collections at these institutions, enhancing the utility of microorganisms in industries. And we know that for the application of any organisms, we need to have its authentic culture in a culture collection. Then only one can develop it into technology. His research has been groundbreaking, especially in identifying novel compounds from fungi with potential applications in treating various diseases. He has led to his research has led to 8 patents, but also some more than 155 publications and 21 books, underscoring his contribution to scientific knowledge and innovation. Dr. Deshmukh has contributed to scientific community through his leaderships, including he served as past president of Mycological Society of India. Currently, he continues to lead and inspire as president of Association of Fungal Biologists and advises several biotechnological companies, showcasing his enduring commitment to mycology and biotechnological innovation. His legacy is a testament to the power of dedicated research and leadership in advancing the science. I welcome Dr. Deshmukh sir for your talk.
Thank you, Shinoy and Rohit. I'll just share.
Yeah.
Is it visible now?
Yes, sir. It's visible. Yeah.
Yeah. So the topic of my talk is natural colors from endophytic fungi for food applications. Actually, this work partly done at Piramal Enterprises Limited, followed by Terry Deacon Biotechnology Center at New Delhi. If we talk of natural colors are dye or pigments which have its original form originated from natural sources like vegetables, microbiological sources, animal and mineral sources. Color of the food is associated with the flavor, safety, nutrition value, and market sale of the product. If you look for the advantage, no toxic accumulation in consumer, safe for all age group. And most of the natural pigments have some health benefit like they have antioxidant, antimicrobial, immunomodulator properties. Food colors from natural sources without any addition of chemicals are approved easily. This work is already published by in Frontiers as well as in one book chapter. If you look for global relevance, globally contribute factor for rising in natural colors demands includes growing concern about the environmental hazard in the production of synthetic food color, increasing awareness about the harmful effect of synthetic colors, rising awareness regarding the advantage of food color, rising number of food industries and offering a new food product, growing concern towards the health benefit. If you look for the global need, the global natural color market was valued in the US dollar 4.6 billion in 2023 and expected to rise US dollar 6 billion by 2028 at the rate CAGR of 5.4%. If you look for North America food colorant market, it is projected to grow at 4.7% CAGR during the forecast period of 20 to 25. In Asia Pacific market will be around CAGR of 4.8% during the period of 24 to 29. Europe and Japan banned trading of synthetic color in food and shifted to natural color. If you see the challenges, these pigments are associated with certain challenges like stability, shelf-life, formulation. High doses are required for the desired hue. Natural food colorants may react on different food metrics differently, causing unwanted flavor and odors. When we started, there were specific objects. First, the identification of fungi for coloring producing property. Then structural validation of the coloring compound, toxicity study for GRAS, means generally regarded as SEF, and optimization of color production conditions and scale-up properties, and finally, nano application of nanotechnology for stability purpose. Objectives are mentioned here: 1, 2, 3, 4, 5. Criteria were: first, identification; second is the identification of the active compound, negative toxicity, nanoformulation with enhanced property, and cost-effective upscaling protocol for the industrial interest. This was the basic protocol: isolation, screening of color pigment, which are not showing, we discard or keep in collection; color producing culture, their characterization, isolation of color compound, structuralization, toxicity studies, optimization of metabolites, and finally nano intervention. If we put in pictorially this one, then these are the cultures what we got: biotechnological tool for metabolomic techniques, genomic techniques for the enhancement of production, or unique Cas9 technique for the removing of the toxic compounds, then is host specific genetic technique, then there will be optimization, large scale production in fermentation, then whatever extracellular or intracellular, they go for extraction either by ultrasound assisted extraction, pressure liquid extraction, microwave assisted extraction, supercritical liquid, ion liquid, or pulse electric field assisted electration. So these all were tried differently on different cultures. It vary from culture to culture. It is easily extractable. We just use the solvent. If it is not, then we try dye and resin. If not, then we try all other techniques which is mentioned here. Then finally, we look for the large scale, nano scale, and look for the marketed pigment. For isolation, as usual, fresh leaves, wash with water, known to all of you. We wash it with ethanol, sodium hypochlorite, ethanol, sterile water. We cut it in small pieces, put agar medium. Here, normally we use half strength PDA plus plant extract from where we got the leaf. Then isolated colonies we purify and use for the color pigmentation. Only in the control, if we are using the leaf, then we wash the leaf, put one side on one plate and other side on other plate. If something is coming, it means the leaves are not sliced properly, and whatever cultures are coming, we don't consider them as endophytic fungi. These are some of the pictures of pigment producing fungi. You can see dark pigment, red pigment, which we are interested. Here the color was little brown, but we were interested more in red because red and blue is more in demand as far as the food industry is concerned. These are some of the identified culture Telleromyces when we grow in the liquid media. After 4 days, this is the condition, and 7 days, and after 7 days, autolysis starts. So we stop that based on the estimation. These are some of the pigment producing fungi, a large number. You can see large number of fungi producing the pigment, but we have to see whether we can get some compound which is of application for particular indication. Workflow, simply as mentioned earlier: colony, this will be put on flask, extracted, color crude, then we will do the TLC column. If needed, we do the HPLC, and then we get a pure compound. For large scale fermentation, if you have more cultures, and then we use this manifold, and in this manifold we can take 20 cultures at a time and check what type of metabolites are produced by the cultures and whether it has any particular pigment of design. This is the flow of the isolation of the compound, isolation and characterization. We take a culture, we put 3 different solvent, then whatever spectrum coming, whatever positive fractions, it go for chromatography, then again spectrum analysis, then HPLC and other things are done. We get a pure compound. Pure compound go for a spectroscopical analysis, and then we'll see the structure. When we are doing the structuralization, we also see whether the compound is known or unknown, and then it go for toxicity assay, fermentation, and other epigenetic modification, and whatever comes, it go for the nano formulation. So this is the chromometer which we use for normally checking the hue of a particular extract in comparison to the carmine, which is a insect pigment, and having the aluminium, so people are not preferring that because aluminium is in the core. This is the chromometer where we can check if it is a red, it is A plus, green is A minus, then yellow is B plus, blue is B minus. With this particular chromometer, we can make out what percentage of red, green, blue, and yellow color is present in our extract. And we compare this with either beetroot and or with the carmine. And this is the formula what we use for the calculation of a color. This is the selection criteria. We check the molecular identification of a culture, then we look for the quality of a color. If quality of color is not desired, we reject them. Then finally we go for the pigment per percentage. Then we look for the food model system, whether it is applicable. Then we use a different media for the production of this particular compound. Whatever suitable media we put, we grow them in particularly in a flask with light extended clay aggregates. So whatever we are growing onto this, if it requires solid, it will be having solid, semi-solid media, and we get a better quality of color with minimum loss. Some of the pigments are carotene, canthaxanthin, astaxanthin, lycopene, toluene, phycoerythrine, riboflavin, and so on. So these are the common pigments what we get with fungi and other microorganisms. If you see the effect of different media, we have tried different media like potato dextrose agar, maltose agar, severots agar, cornmeal agar, the pegdos agar with yeast extract. You can see in some media there is a huge production of the pigment. In some cases, like severot dextrose, there is no production of the pigment. So this depends on what type of substrate is needed for the growth of that, and what condition it is producing the pigment. Then second culture, that was xylaria. You can see it is producing different media. Other is monescus. We have tried similar type of experiment and check whether we can get the better media for the production of that particular pigment. Then we tried the effect of the light as well as dark onto production of the pigments. Here you can see this is the light. Here the slide shows dark. In the case of dark, it produces maximum pigment. So after 26 days, we have tried up to 6 weeks. So after 26 days, it shows better pigment. Then their autolysis starts. This is a spectra of xylaria culture. In dark, you can make out this is the maximum production of red here, and this is the 1 to 7 days data. Similarly, if you look in the light, 16-hour light and 8-hour dark, the percentage of production is a little less. So we have considered the product we have considered that grow the culture into the dark in liquid media. Then whatever extract came, we have extracted with basically dichloromethane, and then whatever came within that TLC with dichloromethane, 1% methanol, 2.5% methanol, 5% methanol. So wherever we are getting like this clear band over there, we do the silica column, and then we get a pure compound. Then this purity of this compound is checked in the HPLC. Large amount of the compound can be isolated using UPLC MS method. Here you can see the spectra of 1 to 1% aqueous solution, sample 1%. So this is the spectra of the compound compared to carmine. So it shows not as good as carmine, but it is showing very good spectra as far as the crude is concerned. Then we have done the LCMS. So this is the peak what we got 1. In the case of LCMS, another. So we have got so many peaks when we are enhancing that. So initially when we see a single peak. When we are enhancing it, so we get so many peaks over there. So then we have done the LCMS here, and then you can see different peaks are present over there at 333, 233, then 1548. So these peaks were present over there. So this is for one. This is for the second extract. This is the mass data. So what finally we got from xyleria is that we have got two to three molecules. I am not showing the structure because it is not yet published. It is already communicated, maybe coming soon. So this was the xyleria isolated from the dhatura metal, and the pigment was red. What happened? Why we have taken this? Because we are seeing this. People were trying the dhatura for various worship purpose, and we were not knowing exactly what for people are producing. I personally felt that is a poisonous thing. So having antioxidant activity. So one was antioxidant activity used for religious purpose, and we have collected this from the Terry Deacon Nanobiotechnology Center at Gurgaon, New Delhi. Another was the fungus producing the pigment was Cephalotheca faevoluta. This was source was Eugenia jambulina. Compound was sclerotin. This we isolated from the our Piramal campus as Goregaon. Here you can see this is the chlorine present over here. But when we are modifying the medium with other solvents like bromide and iodide, we can replace this chloride with bromide and fluoride, but we could not succeed in iodide. Another important culture was the Phomopsis, which is producing altosolanol A, isolated from Lichnanthus arboreus, collected from the Thane, and compound produced for altosolanol. The beauty of this culture, when we are incubating it in the light, it was giving white mycelium. Sorry, when we are incubating in dark, it was giving white mycelium. But when it is exposed to the light, it was giving the crystal. Basically, what happened that we thought this culture is inactive, and we have discarded on Friday, Saturday, it was in the washroom. Monday, we saw some pigment is producing. When we take out the crystal out of it, you can see fine crystals over there. We have got the crystals out of it, so it was producing the ultrasolanol, its various dimers, and its various analogs of this. So we have got the culture, means we have got the compound from this ultrasolanol A, and we have published this. Another one was the oxynthan conjugatum. We have got this as an endophyte as well as from the soil of Khardungala top and Ladakh. Compound was isolated with oxynthan A and oxynthan B. Actually, when I was working with carotenophilic fungi long back, we have isolated this from various parts in the soils of Madhya Pradesh as well as from the Jaipur and other parts. So first we thought it is a carotenophilic, but we have 2-3 times we got from as an endophytic fungi. So I really don't know whether it is endophytic or it is soil, but we got two pigments from here, and it was showing very good hue. Another was from hypoxalon. We have got these three known compounds from the hypoxalon fruit body. Then we have got on monoscus. These two compounds, monoscus-like pigment and hydroxyanthraquinone, both were known, were isolated. Then what happened that whenever we are working, we were getting all known compounds, and yield was very low. So what we thought, can we try some other methods for the production of these compounds? So we thought that we could try epigenetic approach for metabolic diversification. Epigenetic modification brings changes in gene expression without any alteration in DNA sequencing of fungi. Here we are using some chemicals like HDAC or DNMT inhibitors, which induce the formation or which is stimulating the transcription of annotated or silent biosynthetic gene cluster, thereby resulting in the production of a diverse metabolite which is not expressed, or enhancement of variety of secondary metabolites. This is the normal process: transcription, translation, biosynthesis. These are some of the DNMT inhibitors like 5-azacitidine and others, histone HDAC inhibitors like sodium butyrate, sodium hydroxyamic acid, and so on. So these are some of the HDAC inhibitors and DMLT inhibitor what we used in our experiments. This is an example as shown earlier, Cephalotheca faevoluta producing of this sclerotin. When we added 5-azacitidine into the culture. So this is the production extract site sclerotin when initially without 5-azacidine was done. This is the yield. When the similar condition, when we added 5-azacidine, the peak enhances manifold. Along with that, we got additional peaks. So we have got additional peak. We have got a large amount of this, and we have taken the spectra. All these were the new compounds, and this may be coming sometime in Journal of Fungi. Then this is an example of epigenetic modification, but we were facing lot of challenges, and the challenges are low shelf life, high doses are required for the desired hue. This also increase the cost. Natural food colorants may react on different food metrics differently, causing unwanted flavor and odor. Stability against pH, temperature, heat, oxygen, moisture content. Weaker tactile strength. Then we have other environmental related problems were also there, like effect on protein, metal ion, and organic compounds. So these were all sensitivity to different environmental conditions were there. So what we have tried some nano intervention. Okay, before that I will tell you what are the major problems, like what are the major problems associated with the particular carotenoids: anthocyanin, chlorophyll, and beta carotene. Carotenoid is sensitive to light and oxygen. Anthocyanin, effect of pH, metal ion, light, temperature, oxygen, enzymatic activity. Chlorophyll, enzymatic reaction, acid, oxygen, light. Beta carotene is the solubility issue. Turmeric and annatto, there is the solubility issues. So if you talk of annatto, this is a organic red compound which is produced by the ecote tree of which is native of the Mexico to Brazil. So this turmeric and this other one is very important as far as the colorant and as well and other properties concerned. So what we do, we have tried microencapsulation and nano formulation of natural color. What are microencapsulation? Encapsulation is a technique wherein the active compound is mixed with the carrier. The active compound can be flavor, drug, pigment, anything. Possible carrier agent is dependent upon the active compounds, technique used, and reason for encapsulation. Sugar, protein, polysaccharides, gum, and can be commonly used as encapsulate material. Microencapsulation differentiate, we are using here the microparticles, and other things remain the same. If you look for the main principle of microencapsulation, one is colorant, then there is the wall material. So here you can see colorant is there, then one wall material. In some cases, it is in the matrix, it is the dotted form in the wall material. If stability problem still exists, then we put two wall material over there. So our colorant will be inside, then wall material 1 and wall material 2. This is called multi-wall. This encapsulation material, active ingredient, encapsulated matrix. Core material can be solid liquid. Matrix can be polymer, wax, resin, protein, polysaccharides. Vehicle can be aqueous or no aqueous. Main objective for encapsulation, this increase the shelf life, protect the material from undesired environmental conditions, reduce the transfer rate of the core material to the outside environment, modification of physical characteristics of the original material for easy handling, tailoring of release of the core material. This is important. To suppress the unwanted aroma test of the core material, to separate the component from matrix that could easily react, to dilute the core material when it should be used in a very small quantity. What can be the coating or matrix formation, including several categories? Wax is one, protein is second one, carbohydrate, and food grade polymers. Wax and lipids can be beeswax and so on. Protein can be gelatin, soybean protein, gluten, and so on. Carbohydrate can be maltodextrins, chitosine, sucrose, glucose, starch. Food grade polymer can be polypropylene, polyvinyl acetate, polystyrene, polybutyrene, and so on. So these are the metrics used. If you look for the wall metrics and other information, maltodextrin is one, exhibit high water solubility, low viscosity, blend flavor from colorless to colorless solution, lack of emulsifying property, and poor flavor retention. So these are some of the properties which maltodextrin have, some limitations. Modified starch is another one, good emulsifying property and stability for emulsion formation due to its addition of octanyl succinate. So these are some cyclodextrin, gum arabic, inulin, chitosan. Chitosan mostly we have used. This is lyophilic encapsulation, have various bioactivities, biological, reproducible, non-toxic, biocompatible, and biodegradable. Other one we have tried was gelatin. It is a good emulsifier, film formation, water solubility, edibility, and biodegradation property. So mostly we have restricted to the chitosan, gelatin. If you look for wall material, their property soluble in water. If you see nothing, alginine, mechanical. So these are the wall property and release. What is the release mechanism of all these compounds? Then what various methods we have tried for the microencapsulation process in food industries. What we have tried is highlighted in blue, spray drying. Other people are trying spray chilling, air suspension coating, spray cooling, fluoride bed coating, centrifugal extrusion, freeze drying, coacervation, co-crystallization, liposomal entrapment, interfacial polymerization, molecular inclusion, and emulsion. So whatever highlighted with blue, we have tried those along with the liposomal entrapment. Here, what is spray drying? Core material is homogenized with the wall material in a suitable solvent. Homogenized mixture is then fed into a spray dryer and automatized with the nozzle or spinning wheel. Water is evaporated by the hot air condition to anatomize material. Capsules are precipitated to the bottom of the collector. In modern spray dryer, viscosity of solution to be sprayed can be as high as 300 millipascal second. So we have used both this users. One is nano B90 and mini spray dryer B290 from Bushi Laboratory AG. Both are the glass one, and our size was small because we were having limited number of the red pigments available with us, limited amount. Maybe it was not more than 1 gram or 2 grams in all the cases. The advantage of this is possibility of employing a wide variety of encapsulating agent, applicability of both the hydrophilic and hydrophobic polymer solution, potentially large scale production, simple equipment, good efficiency, reduced storage, transport cost, and low process cost. Disadvantage, loss of significant amount of the product due to sticking problem. Possible degradation of sensitive product at high temperature because the temperature reaches up to 300 degrees Celsius. Fiber formation if wrong wall material is selected. So these are some of the disadvantage. Next one was freeze drying or lyophilization. Simple process. In the freeze drying, the material is then reduced the surrounding pressure and adding enough heat to allow the frozen water in the material to sublimate directly from solid phase to gas phase. So basically it is sublimation process. Help in encapsulating water soluble extract like essence and natural aroma as well as drug. So this is basically sublimation process using the particular technique. Advantage is this is useful for thermosensitive substrate and it is used for unstable in aqueous solution. Disadvantage, it is 50 times higher cost than the spray drying. Storage and transport of the particles is extremely expensive. Commercial applicability is severely restricted by a long processing time. So this, if you put any material for spread freeze drying, it takes around 48 hours to 48 hours to 74 hours. Another method, what people were trying, were co-acervation. This is a phase separation of one or more hydrocolloid from the initial solution from the effect of damage of pH or temperature, addition of non-solvent or electrolytic compound. The newly formed coacervate phase deposits around the active ingredient suspension are emulsified in the same reaction media. There are three steps involved. First, you see here is the one. Then in second step, it is depositing on that particular material. In third step, it is basically this is coating the outer layer, and lastly we rigidifying the coating. So by this method we get the emulsion nanomaterial, and this method is little cheaper also, and if it is working, it is economical also. Advantage is relatively simple, low cost, does not require high temperature or organic solvent. Disadvantage is limited with a narrow range of pH, colloid, colloid concentration, and electrolyte concentration. It is called simple collaboration when only single micromolecule is present. Whenever there are two or more molecules of opposite charges referred, it is a complex coacervation. Another one is emulsion. Emulsion, encapsulation of bioactive in aqueous solution which can be used directly as a liquid or dried to form powder. Include two immiscible liquid, usually oil or water. One of the liquid is dispersed as a small spherical droplet in other one. Oil droplet dispersed in aquatic phase, oil in the water emulsion. If water droplet dispersed in the oil phase, then water in oil emulsion. Mechanically, energy disrupts the two phases to an extent that droplet from these large droplets are subsequently broken down into small one. This is critical step in emulsification and encapsulation. This is also a simple one but required skill. Simple and cost-effective method. Aqueous phase is easily flavored. The oil sensation is easily removed. The rate of absorption is increased. It is possible to include two incompatible ingredients, one in each phase of the emulsion. Disadvantage, the emulsifier has to be chosen appropriately so that it does not interact with the food matrix. Preparation need to be shaken well before use. A measure device is needed to be administration. A degree of technical accuracy is needed to measure the dose. This is very important that one has to be very careful. And last one is the storage condition may affect the stability. So when we do a small scale, we also see the storage condition needed for this particular type of encapsulation. Then another one is nanoemulsion. Nanoemulsions are the emulsion with droplet size on the order of 100 nanometer. A typical nanoemulsion contains oil, water, and an emulsifier. The addition of emulsifier is critical for the creation of small size droplet. Emulsifier decreases the interface tension between the oil and water phase of the emulsion, stabilize nanoemulsion through impulsive electrostatic interaction and the steric hindrance. The emulsifier used generally is surfactant, but protein and lipid have also been used. But some people tried, but we have used very limited number of proteins. Nanoemulsion compared to a micro and macroemulsion, larger surface area per unit volume have strong kinetic stability and less sensitive to the physical and chemical changes. If you see this, then this is the process. Organic phase, put in aqueous phase, bring phase together, then water diffusion, micro, then microphase breakup, then microemulsifier, emulsion form. So these are the process used, and this is the simple process, but it takes very specific technique or specific what you call conditions for forming the nanoemulsion. If it is not tried, then it may your experiment may fire back to you. If you compare between microemulsion and nanoemulsion, macro is size is 1 to 100 micrometer, micro is 2 to 20 to 500 nanometer, and nano is 10 to 100 nanometer. So this is micro, nano 20 to 500, 10 to 100. Spherical, or laminar in the case of nanoemulsion. Stability, thermodynamically unstable, weakly kinetically stable, thermodynamically unstable but kinetically stable, and nanoemulsion is thermodynamically stable. And kinetically also it is stable, but we have not done much work on that. Method of preparation, high and low energy method, high and low energy method. This is low energy method. Then polydispersibility often high, then it is medium, and in the case of nanoemulsion, typically low, it is less than 10%. We have tried with the known methods for beta carotene by Yuan et al. They have used the size and stability of nanoemulsion with beta carotene against temperature, pH, and surfactants. Because this was our first experiment, so we have followed with this method. Again, followed the method of Quinn et al., where beta carotene and stabilize them with the lactoglucan, a biocompatible emulsifier. Beta carotene nanoemulsifier have been prepared with different methods like light pressure homogenization, microfluidization, and evaporative ripening, and different emulsifiers. So this we have tried after using these two methods which were tried earlier, which were already known in the literature. If you look for the example of microencapsulation applied for food colorant, then astaxanthin, chitosan, gum arabic, proteins, maltodextrins, inulin, glucose. So these are all list of the raw material used. People have tried spray drying. Most of the people they try freeze drying. Some use the inotrophic gelatin spray drying and freeze drying. So these are the compound, different colorants, raw material used. These are the colorant, raw material used. These are the technique used. These are the various references where we have collected this data before starting our work of the nano encapsulation and micro encapsulation of these food colorants. If you look for food application, nano encapsulated color can be used commercially as a food dye. Possible food material could be yogurt, ice cream, and so on. In literature, food application of microencapsulated natural colorant have been investigated in several studies. These are few examples where anthocyanin, raw material metrodextrin for yogurt and salt drink, red beet extract, this raw material of maltodextrin used for yogurt, beta-cyanine, glucose syrup, lycopene, modified starch used for yogurt and soft drink. These are the references. This is just to show that people are trying our this core material and mono material used as an encapsulated material. If you look for the endophytic fungi can be a potential source of natural pigment. Natural pigment can be produced in a controlled condition. Natural pigment can provide further health benefit like having antioxidant, antimicrobial immunomodulator activity. Nano intervention can challenge help in challenges faced in natural color and control. These are the acknowledgement. Professor Colin Barrow, he was the guide for some of the students when I was with the Terry Deacon Nanobiotechnology Center. So he was the main guide, I was the co-guide, and we have done lot of work with him on the bioactive metabolites as well as colorant from the fungi, particularly. Shilpa Verekar was with us in Piramal Enterprises when we are working some initial work. Shivankar Agrawal is Scientist D at Indian Council of Medical Research, New Delhi, and Tanuka is the Oxford University postdoctoral fellow. So their contribution in the pigment production is really commendable. Tanuka's paper, we have two papers she has published on this. One is in Frontier, which is around 250 citations. Shivankar has published more than 10 to 15 papers on bioactive from this. So these people have contributed significantly for my work. This is the place where I worked last at Terry Deacon Nanobiotechnology Center team. So this is the team, my team where I was working. This is the center, Terry Deacon Nanobiotechnology Center. This is the when I was the president of Mycological Society of India. These are some of the books edited by me and our different colleagues, including Professor Sridhar, Professor Satnarayan, and others. At the last, I would like to highlight some points about the Association of Fungal Biologists. The basic objective was to create awareness about the fungi and their application in biotechnology, because what I was feeling that we were working more on the taxonomy and less on the application in biotechnology. What we are doing, we are organizing the webinars, awareness program on various platforms. This helps the youngster in collaborations. We have around 175 live members, expected to reach 200 in 2024. Interested people can become a life member or annual member by contacting me or AFB. We had the first general body meeting held in December 23 at Mumbai, SIS College. Second AGM will be held in October 2024 in Pune. We will be starting a new journal soon covering wide aspects of fungal biotechnology. So these are some of the idea about the Association of Fungal Biologists. And at end, I would like to thank MycoIndia as well as MycoAsia for collaborating with us. This is sort of mutual understanding among us to create awareness about the fungi and their application in biotechnology. At last, I am thankful to Belle Shenoy, Rohit Sharma, and others for giving me a chance. Thank you very much.
Thank you very much, sir. Thank you very much for that wonderful presentation. I hope I am audible to you.
Yeah.
Yeah, thank you very much for that wonderful presentation. I echo your sentiments that we need to create more awareness about the applications of fungi in our normal day-to-day life, also in industrial setup. Thanks for highlighting that point, and also I agree with you that the Association of Fungal Biologists has been wonderful in terms of creating awareness through webinars and all, and I would like to see more members joining that Association of Fungal Biologists. And I will let the audience ask questions. Mr. Arvind, do you have any question?
Yes, sir. Thanks for the opportunity, sir. So it was a wonderful presentation, sir. It was very grateful to see the pigments in the fungus. With my first question, why does this fungus produce these pigments, sir? Like, what is the use for them, sir, producing this pigment?
Basically, they are producing for their survival. I will give one example,
Okay,
which is the example of oxynthron conjugatum.
okay, sir.
If you look for morphology of oxynthron conjugatum, it is having the appendages onto the clusterothecia. And
Okay, sir.
this is dichotomous, which help us in attaching to the feather of a particular bird.
Okay.
And the pigment is yellow. So what we thought, means what is our hypothesis, that this pigment is protecting the ascospore of that particular fungi from UV radiation. So this may be one reason. Secondly, they may be producing for their own survival.
Okay.
Because they have antimicrobial property and other biological properties, so they may be producing for their survival as well as for the killing the other microorganism which is harmful to them.
Okay. So with the second question, you said about this pigments will be used for the food processing material. So in terms of food processing as an application, to study the toxic study, so what type of cell lines will be choose, sir?
Normally, we look for the toxicity of particular extract. I will give an example of particularly the moniscus, where
Okay.
we were getting the citrulline production over there.
Okay.
Citrulline is a mycotoxin. So using the
Okay.
Cas9 system, our scientists have removed that, and we were having citrulline-free thing.
Okay.
One, that is one.
Okay, sir.
Secondly, if we are using, then we use the normal human cell line,
Okay.
WI38, at
Okay.
little higher concentration to check whether it is toxic to the human body or not, human cell line or
Okay.
not. And
Okay.
then we try the animal experiment also to check whether it
Okay.
is giving any side effect.
Okay. Sir, again, the third question: whether these pigments are synthesizable, sir, artificially in the lab, as being a natural one?
Some can be synthesized, but not all.
Okay. Okay, sir.
Because for
Tha-
synthesis also you have to involve manpower over there, and
True,
it is
sir.
a big task. If you want to
Okay.
synthesize one culture, one should be very careful while taking the synthesis work, because money is involved, and what is the
Okay.
applicability after synthesis.
Okay. That is also.
Anything else?
No, sir. Thank you very much, sir.
Thank you. I love this enthusiasm for asking questions, getting the expert opinion. But I request the audience to just limit to one question. Maybe if you get a chance in later part of the session, you can ask. Ravi Ramanathan, please ask your question.
Yeah, thank you. Good morning, everybody. Sir, wonderful presentation. I'd like to, you mentioned a couple of times about the yield of such pigments. So what about the yield? Are they high or are they low? That's one question. And the other related one is about the scaling up. So you have not talked about how to scale up the production using these microbes, these microbial fungi.
See, basically what we saw first time, then when we are getting the yield is around 5 to 10 milligram per liter. Normal condition, what we are seeing, 5 to 10 milligram per liter. But when we modify the medium and the other conditions, it reaches up to 200 to 300 milligram per liter. So this is what we have done in the lab condition. But if you do the genetic modification, it may go up to 500. But we have tried this for very limited number of culture, not for all. So what we reached up to 500 to 800 milligram per liter. But if culture is of some interest, genetical modification can be done and can be increased more.
Okay, sir. Thank you. This about the scale up, because fermentation in larger tanks is required, and purification at that scale is also required. So TLC kind of things won't work.
What we were doing, we were taking we are taking out the extract at different interval, and we are using the chromometer to check the hue, red color particularly. So starting from 48 hours till almost 7 days, we were trying. And maximum in the case of moniscus, we got after 6 days in the 25-liter fermenter. So that way we were trying. We are not using HPLC, we are not using TLC, but extract we are trying with the chromometer first, and then we are also trying with the HPLC to see exact amount what is produced.
Thank you very much, sir. Thank you very much.
Dr. Shishipala, please.
Sir, namaste.
Namaste.
Sir, quite interesting work. One thing which was bothering me since we were working on pigments, many of the pigments, fungal pigments, identified using LCMS and related software.
Yeah.
The metabolites are of common origin, which overlaps with the plants, plant
See,
pigments.
uh-huh.
Which is an indication of phylogenetic relationship between fungi and plants with respect to pigments.
Actually, I have not worked on this aspect, but my thing is that whatever pigments we got, we have got from the fungi, and they are not produced by the plant particularly. And
Okay.
of late, we are focusing on the marine system because we felt that it is a tough system, and our rate of production of this what you call
Yes.
hit rate was much higher in compared to other other endophytic fungi. So we were trying all with the mangrove and soft corals and others. So where the
Okay,
hit
sir.
rate
Okay,
is
sir.
high.
Thank you, sir. Thank you.
Dr. Chandan, please.
Thank you, sir, for giving the opportunity. Sir, my question is, I want to know whether Ganoderma is endophytic fungi according to you.
See, I have never got Ganoderma as endophytic fungi, so I cannot comment on that. We have got only Ganoderma as a parasite, particularly on this plants, some plants around the garden and others. I have
Okay,
no
sir.
idea.
Sir, in your slide, I have seen that you have mentioned Giardia species as endophytic.
Yeah.
And we study Giardia as one of the parasitic or is a saprophyte fungi like a mushroom. We treat in a mushroom category, sir.
See, Xyleria you can get on the wood, rotten wood.
Yeah,
Xyleria you can get
yes,
associated with the termite nest. Xyleria
yeah.
you can get as a endophytic fungi and so on. So what we got from the particularly from the mangrove collected from the Alibag near Mumbai.
That means you have got from the live plant, sir.
Live plant, yes.
Okay, sir. That was why I was confused with that slide.
Hey, even
Thank
if
you.
you see the dead wood, you will get the Xyleria. There's no
Yes,
problem.
sir. Yes, sir. We have reported that one. That's why I have asked the question. Thank you, sir. That's all. Thank you.
I love this enthusiasm. Once again, please limit to one question because there are 5 or 6 more people who are interested to ask questions. I think it's fair to give a chance to all. Thank you so much. Dr. Mohan Kumar, please.
Thank you for this opportunity, Vile sir. Deshmukh sir, I have a question. What makes this endophytic fungi to produce? Because endophytic fungi mostly it is present inside the plant and is dominated by the plant pigment. But
Yeah.
why it needs to produce? And is it the same with the soil isolate or any other isolates of fungi?
See, when I was working with the endophytic fungi for particularly production of the bioactive metabolites for antimicrobials, anti-cancer, anti-inflammatory, that time we were getting large number of pigment producing fungi, which were producing red, green and others. So reason I do not know why they produce, but they were producing to be concentrated on that because our company were interested also in food.
But
And
but chlorophyll is rich in that, but we don't see any that pigment is dominating in the plant species.
see, I have no idea.
Okay, and the micro encapsulation has been done for the fungal pigment.
Yeah, we have tried with the means monoscus pigment. We have tried with the telomyces pigment and oxynthron pigment. Along with some little work was done with the altosolanol A. This we have done with the collaboration of Deakin University, Australia, where Professor Colin Barrow was our chief investigator.
Okay, thank you, sir.
Mansi Verma, please. Kaushik Bihar, please. Professor Sridhar, please.
Hello, hello, Dr. Deshmukh.
Yeah.
Good evening.
Good evening.
You are in New Zealand now.
Yeah.
Now, can you please highlight something about the pigment produced by some of the macrofungi, especially with regard to melanin is concerned? Can you highlight something? It is very useful, I think.
See, if you think of melanin production, mostly what we saw, mostly it is in the east, particularly what east we got from the Marion. But melanin is also produced by some Aspergillus, Penicillium and others. Even by some what reports are there from Agaricus and others.
Okay, yeah, including termitomycetes.
Termitomyces.
Yes,
Even
yes.
some melanin is produced by the cordyceps and others.
Any usefulness for food industry?
What I know is mostly they are using in cosmetical purpose.
Okay. Thank you very much.
Thank you, Professor Sridhar. Samita E.
Sir, I have one question. So essentially, how long it took for the method development to analyze these compounds by HPLC, sir?
See, it depends on the complexity of your extract. Sometimes what happens that if only one compound is produced, then you may get idea in 2 to 3 weeks time when you get the extract. If it is a multiple, then you do the repeated HPLC and UPLC, then may take some time, 2 to 3 months time. It depends on the expertise of the chemist involved in that.
Okay. So, sir, so we have to have more samples before you start going to these experiments, correct, sir?
See, normally we start with 500 milligram to 1 gram of the crude extract. So to get about 20 to 25 milligram minimum compound for the structural mutation and others.
Okay, sir. Thank you, sir.
Nalini Somaya, please. Priti Naik, please.
Sir, out of all the encapsulation techniques that you have discussed, which
Yeah.
is the most preferred industrially used one? I was very intrigued by the encapsulation techniques actually. So, if you consider industrial application of these encapsulation techniques, which is the one that is mostly used because most of them have their own advantages, disadvantages. So, currently, which is the one that is used?
See, cost effective and better is the spray drying, and only disadvantage is that one has to try, means at least first 2 3 times you have to try, and then it works consistently. So spray drying is the best one.
Okay, sir. Thank you.
Kaushik Bia, please.
Hello, sir.
Yeah.
My question is that you said these pigments have antimicrobial property.
Yeah.
Does they taken as food? Do they infect gut microbiota?
See, I have not tried that. I have no idea.
Thank you, sir.
Stands in Dulma, please.
Thank you, sir. Sir, my question is like, can fungal pigment also used for dye colors? Because in Ladakh, I have seen lichens being used as a dye to dye for color, but not fungi.
See, if you look for the lichen, it is association of yeast, fungi, and algae.
Yes,
So
sir.
you have to see which is producing that particular pigment. And lichens are used for the colorant since civilization time. People are using this for making paintings and others. So Ladakh, particularly those tribal people, they are using some of the culture, some of the samples we got. They are produced by the particularly by the fungi, as I told the example of oxynthron, which was
Yes, sir.
actually isolated from that some tree as well as from the lichen. So really, which part of that particular means what partner is producing, that we do not know.
Okay, sir. So can fungal pigment also be used as to dye colors?
Yeah. People are using for coloring. One Dr. Rice was there. She has published large number of papers on the production of dye from the mushrooms, from other fungi for textile industry.
Okay, sir. Thank you so much, sir.
Thank you very much, ladies and gentlemen. Our next speaker is already here, and I don't want to keep him waiting for this chance.
Sir, I wanted to ask a question. May I please, sir?
No, madam, sorry, we don't have time
Okay.
right now. Thank you.
Okay, sir. Thank you. Thank you.
So, Professor Dr. Deshmukh, thank you very much for your spending your time and collaborating with MycoIndia, and of course sharing your experiences with these issues. And I'm sure many people will have many more questions. And Dr. Deshmukh can be contacted on his LinkedIn, Facebook, WhatsApp, and email also. I go over to Dr. Rohit Sharma for the final comments, please.
Thank you, Dr. Shanath. Thank you, Dr. Deshmukh sir, for a very nice and informative talk. I think related to the biotechnology of pigment, I think it's you covered a lot of things, including the extraction process, steps involved into it, fermentation conditions, and even some aspects related to the large scale or scale up kind of things. And it was amazing to know that even the endophytes, apart from the antimicrobial activities, it was amazing to know that they have the potential source of pigment. You also mentioned that the challenges faced during the same, and also very nice information related to the micro, macro, and nano emulsions. I think. And as you mentioned in your talk, that spray drying is the most popular technique that people are using for this thing. And I think people working on pigments will now be able to take their work to a conclusion level using the flow of processes that Dr. Deshmukh sir has mentioned on pigment research and technology transfer also. So I suggest that so watch out for your pigment producing fungi before discarding any culture petri dish now onwards. So thank you, Dr. Deshmukh sir, for a very nice and informative talk. Thank you so much.
Thank you.
So moving to our next talk, our second talk is by Dr. Bhupesh Taneja. So